Hi, my name is Matt Jenkins, and I am a senior at the University of Tennessee, double majoring in neuroscience and microbiology. I work as the lab manager in Dr. Cooper's behavioral neuroscience lab, where we use a Syrian hamster model to investigate stress and stress coping mechanisms. Almost everyone experiences stress, but few people go on to develop stress-related psychopathologies such as PTSD, general anxiety, and major depressive disorder. Stress coping mechanisms can differ between individuals, and one factor that impacts stress resilience is dominance status. The medial amygdala is a brain region that has been shown to regulate aggression and social activity, and this brain region projects to many areas, including the bed nucleus of the stria terminalis, or BNST. The BNST is involved in anxiety and fear-related behaviors. To visualize neural circuits, we use toxin B, which is a retrograde tracer that is taken up by axons and travels up to the soma. It is fluorescent and allows us to visualize neural circuits. To measure activity, a cellular marker of activity called CFOS is measured. Social defeat is used to elicit stress responses in rodents, so we can look at individual variation in stress responses. Previous research has shown that dominant male hamsters show more activity within the medial amygdala and show less social anxiety following a social stressor compared to subordinates. Our objective is to determine if dominant and subordinate hamsters differ in the activation of medial amygdala cells projecting to the BNST. We hypothesize that dominant hamsters will show more activity within this pathway after establishing social dominance and after social defeat stress compared to subordinate counterparts. To conduct this study, we used a resident intruder paradigm in Syrian hamsters. Syrian hamsters are aggressive and highly territorial and will defend their territory once they've established it. Our hamsters are weaned 21 days after birth and then live in a group house setting until 9 weeks when they're individually housed. We individually house them so they will develop a territory and will defend their territory. The animals are then paired with a weight and sex matched conspecific for 5 minutes every day for 12 days. During this time, the hamsters will form a dominant-subordinate relationship. During the middle of these pairs, the hamsters receive a stereotaxic CTB injection into the BNST that we will talk about in just a second. We want to look at how this medial amygdala to the BNST pathway is involved during dominant and subordinate relationships. So one group of the study is sacrificed and has their brain extracted 90 minutes after the last pair encounter. To see how this pathway is involved in stress responses, the other group of animals is sacrificed and has their brain extracted 90 minutes after a social defeat encounter, which is 3 5-minute encounters with a larger, more aggressive hamster that will physically dominate them. We then perform immunohistochemistry to stain the CFOS cells in the brain. The brain sections are then visualized under a microscope. The first image shows cells in the medial amygdala that are stained red. These are CFOS cells. That means these cells are active. In the next image, you can see the green CTB cells. These are cells in the medial amygdala that project to the BNST. The final image is the first two images superimposed on each other, so we can see dual labeled cells, which means the cells are active and projecting to the BNST. In the results section, the first row of figures shows the data taken after the last day of pairs. In the first figure, we're showing the total number of CFOS cells in each brain region. So we have the dorsal and the ventral medial prefrontal cortex. Males are shown in shades of blue, while females are shown in shades of red. The y-axis is total number of CFOS, or active cells. Here we found a sex difference but no status difference. Here we can see that males show more activity in the dorsal medial amygdala compared to females. The next figure shows the percentage of dual-labeled cells over the total number of CTB cells in both the dorsal and ventral medial amygdala. This is showing the percentage of active cells out of all the cells that go to the BNST. Here we see that dominant males have a greater percentage of active cells that project to the BNST in the dorsal medial amygdala. Dominant males are showing around 60% in the dorsal, while subordinates are only around 25%. The last figure in the top row shows the dual-labeled cells divided by the number of CFOS cells. So this is asking, out of all the active cells in the medial amygdala, how many project to the BNST? This lets us know how important those BNST projections are. In all groups, only around 25% of active cells are going to the BNST, which means around 75% of the active cells are going to other brain regions. The next set of figures show preliminary data of male hamsters after social defeat. These have small sample sizes, so more data is needed. However, dominants are shown in light blue and subordinates are shown in dark blue. The first figure shows the number of CFOS, or active cells, following social defeat. It can be seen that subordinates have more activity in the ventral medial amygdala, which goes against what has been previously shown. The next two figures show no differences between dominants and subordinates in the percentages of dual-labeled cells divided by CTB cells or dual-labeled cells divided by CFOS cells. In conclusion, we have shown that males show more activity in the dorsal medial amygdala following daily dyadic pairs than females. We have also shown that dominant males show a greater percentage of active neurons in the medial amygdala that project to the BNST. The results from the defeat animals are counterintuitive from what previous research has shown, so more data is needed. We will also plan to incorporate females into these data as well. And ultimately, we plan to look into other circuits that are involved in stress-related behavior, such as the medial amygdala to the ventromedial hypothalamus. This research is important because understanding stress-related pathways could help better understand how physical changes in the brain affect behavior, and could ultimately be used to better understand stress-related psychopathologies such as PTSD, generalized anxiety disorder, and major depressive disorder. Thank you for listening.